Good afternoon, everyone. I'm Aryan Pillai, and today I'm excited to take you through a topic that is both fascinating and crucial to our understanding of biology and biotechnology. As we dive into this topic, we'll explore key concepts and uncover the latest advancements. I hope to not only share insights but also spark some interesting discussions along the way. The disease I've selected is anthrax disease. Anthrax is a severe infectious disease caused by the bacterium Bacillus anthracis, which can affect humans and animals. The bacterium forms resilient spores that can survive in the environment for long periods, making it a significant health risk, particularly in agricultural settings or through bioterrorism. It manifests in several forms: cutaneous, gastrointestinal, inhalation, and injection, each with distinct symptoms and varying levels of severity. So while anthrax is rare, it can be fatal without prompt treatment, making early detection, effective antibiotics, and vaccines crucial for controlling the disease. When we look into its historical background, it is important to understand its historical significance. Bacillus anthracis was the first bacterium identified as a disease-causing agent, with Robert Koch proving its role in illness in 1876 and Louis Pasteur creating the first vaccine in 1881. As a bioweapon, due to its lethality, anthrax has been used in biowarfare since the First World War, and as a bioterror agent, notably in the 2001 US anthrax mail attacks. Now, as public health risk, aerosolized anthrax over urban areas could cause high casualties, up to 95,000 deaths in a city of 500,000, overwhelming medical resources and infrastructure. Now, epidemiology. In that case, we have to understand geographic prevalence. Anthrax is common in agricultural regions such as the sub-Saharan Africa, South America, Asia, and parts of Europe. Outbreaks also occur in livestock in Canada, the US, and Europe. Human exposure-wise, cases have occurred in people handling contaminated animal products, especially in endemic areas. This includes those who work with animal hides, such as drum makers and wool sorters. Historically speaking, inhalational anthrax, known as wool sorter's disease, was more common in the 19th century among those handling animal products. And for bioweapon research, anthrax has been central to biological weapon programs in countries such as Japan, the former Soviet Union, and Britain for over 60 years. But there has been a decline in cases, with global human anthrax cases dropping significantly from the 1950s to the 1990s. Some high-risk groups include farmers and livestock handlers, military members, and travelers to countries known to have problems with anthrax, researchers, and laboratory workers who study the bacteria, among many more. But there are four different types of anthrax. Cutaneous anthrax, which is known to enter through the skin. It's common in those people who handle animals, and 95 to 99% of anthrax cases are of this type, the fatality being 20% without treatment. The second type of anthrax is gastrointestinal. It's caused by eating undercooked infected meat and affects the stomach and intestines. It has a 60% survival rate with treatment. The third type is inhalation anthrax. Caused by the inhalation of spores, it's common in wool mill workers and is known to cause respiratory issues. It has a high fatality rate, about 50% survival with treatment. The last type of anthrax is injection anthrax. Spores enter through contaminated injections, for example heroin, and is more common in Northern Europe. Some tests we use to detect the presence of anthrax include a biopsy of skin lesions, blood test, chest x-rays, CT scans, lab tests on stool or mucus, a lumbar puncture, also referred to as a spinal tap. But there are several detection and identification issues. Bacillus anthracis shares strong similarities with Bacillus cereus, making it difficult to differentiate, with some scientists suggesting it may be a pathogenic variant of Bacillus cereus. Environmental samples typically contain spores, while clinical samples usually have vegetative cells, again complicating identification. Some conventional methods include selective media growth, lack of hemolysis, motility testing, capsule staining, gamma-phage lysis, and penicillin susceptibility, while modern techniques include DNA amplification methods such as PCR, immunoassays, and biosensors. They offer rapid, sensitive, and portable options for detecting and identifying Bacillus anthracis. Some of the most notable symptoms observed in this disease includes a painless papule that will turn into a black eschar. It's also known as a black skin lesion, while ingestion anthrax is characterized to have a severe sore throat, trouble swallowing, and neck swelling. Inhalation anthrax is characterized as fever, chills, and lethargy, which ultimately progresses into shortness of breath and chest pain. It is important to understand the pathogenesis of this bacterium to be able to understand more about the disease. Bacillus anthracis spores enter the body through broken skin or inhalation, ingestion, or even injection. Inside the host, spores will activate and multiply. The bacteria will then release toxins that cause tissue damage, edema, and immune suppression. The bacteria's capsule helps avoid immune detection, and ultimately, without treatment, the infection can spread, leading to severe complications or death. However, there are many prevention and treatments. The first being antibiotics, which is taken for a duration of 60 days. Penicillin is preferred for inhalation anthrax, but is not effective against all strains. When it comes to antitoxins, injectables such as raxibacumab and oblitoximab neutralize anthrax toxins, not the bacteria. Surgical removal is used to treat injection anthrax by removing infected tissue, while vaccines such as AVA, anthrax vaccine adsorbed, is used against anthrax toxins. There is significant research undergoing right now regarding this disease. When it comes to vaccine research, it focuses on developing enhanced vaccines and evaluating the current anthrax vaccine. Bacterial mechanisms wise, there's research into how Bacillus anthracis obtains iron and evades the host defenses. Genotyping advancements include techniques such as MLVA and SNP typing that have improved strain identification. Finally, immune evasion. Bacteria will use molecular camouflage to avoid immune detection. So I would like to conclude that anthrax is a serious infectious disease caused by Bacillus anthracis, with different forms including cutaneous, gastrointestinal, inhalation, and injection anthrax. So while it can be fatal, timely treatment with antibiotics and vaccines significantly reduces the risk. Continued research on vaccines, treatments, and diagnostic methods is essential to controlling this dangerous pathogen, especially in the context of bioterrorism and zoonotic transmission. Thank you.